All right, so welcome. We are going to be covering chapter 4 this video, which is about cell biology. So let's get the ball rolling. So here are some individuals that were very pivotal in helping us to understand exactly where cells came from. And it's not many names I want y'all to understand, but just a few. It's pretty similar or pretty clear contributions that they have towards the field. So first is Schleiden. He was able to determine that all plants are actually made up of cells. So they're not made up of purely carbohydrates or purely proteins. They're comprised of individual little cells. Schwann, that sounds a lot like the animal swan. He realized that all animals are made up of cells. And then finally, Virchow understood that all cells come from pre-existing cells. So they don't just spontaneously arrive. They have to arrive through cell division. So this introduces us to the cell theory. And if you remember conversations we had about a theory, all theories are never going to be 100% proven, right? We can't go back in time and understand what the very first cell was. We also know that theories are widely accepted. That means we have enough information to believe that these things are true based off of our evidence. However, we can, again, never go back and verify these things. So let's talk about the three components of a cell theory. The first one is all living organisms are comprised of one or more cells. So if it's just one cell, that's a unicellular organism versus more than one, which it would be multicellular organisms. But anything that is living is going to be comprised of one or more cells. Kind of going back to that same concept, cells are the simplest unit of life. So you cannot take anything and break it down smaller than a cell and have a living thing. That's the lowest we can go. All of our reactions in life are going to take place inside of a cell, and cells are going to contain this hereditary information like our DNA, which can make our RNA, which will eventually make our protein. So all of that is going to be housed and completed within a cell. And then our final component of the cell theory is that all cells come from pre-existing cells, and they get here by cell division. So we don't have any random magic where you snap your fingers and a cell pops up. This spontaneous generation does not occur. All cells have to come from pre-existing cells. So this is just an example of some mice coming out of a container of grains, letting us know that even though the mice might have just appeared, there probably was, some tiny embryos present. That's really common with meat. If you let meat sit too long, you will eventually get flies. And those flies are derived from maggots, which are teeny tiny eggs that have been deposited onto the meat. So although it looks like things pop up, all cells are coming from pre-existing cells through that cell division. All right, so another name I want you to remember, and this is probably the last name for this particular chapter, is Robert Hooke. So Robert Hooke was the person that coined the word cell. So prior to that, we didn't really have a terminology for this membrane enclosed area, but Robert Hooke determined that it was going to be called a cell. He was a Mason, and at the time, he was able to kind of correlate the size and the shapes of cells that he had seen under cork or under a tree, and was able to determine that kind of reminded him of like the hallways and the rooms that were in the location that he was living at. So this is going to be another name that you have to keep in mind. Robert Hooke was the person that came up with the word cell. So this chapter does cover information about microscopes, but we will not cover it in lecture. I do want to give you just a little bit of a image to kind of help you. A lot of things we can see with our eyes, but there's many other items, especially biological items, that we need a microscope or some type of tool to see. So this is us as humans. We can see certain types of eggs, fish eggs, bird eggs, that type of thing. But right around about 50 micromillers of space, we kind of need some type of microscope. That's where we're going to be able to see our cells, our nucleus, mitochondria, a lot of bacteria. And then there are even other microscopes that are stronger that allow us to see some things like viruses, bacteria, that type of aspect. Interesting to note that we do not have the technology yet to see things like atoms or even molecules. So we actually do not know what something like H2O looks like under a microscope because we don't have a microscope powerful enough to see something that small just yet. All right, so if this was confusing, if you have any questions, make sure you pause here and rewind the information that we've covered just briefly. So let's talk about that cell biology and what that means. Cell biology is the study of these individual cells, not only the cells, but also how they communicate with one another. So we're discussing different variations of cells, what are the components of most cells, and then also how cells can connect and communicate in your body. What are the types of messages they send? How do cells regulate their environment? Those type of aspects. So again, building off of that aspect of the theory of cells are the simplest unit of life, we know that a cell is the smallest location that we can have a living organism in. And this is an example of a plant cell, which we'll look at in depth. So here, the first thing we're going to do is talk about the two different classifications of cells. Cells can either be split into being prokaryotic cells or eukaryotic cells. So I'm going to zoom in just a little bit. You see a prokaryotic cell is going to be, off the just vision, you can see it's a little more simple. It does have a lot more items on the outside, but on the inside, it's not a lot of moving parts there. However, a eukaryotic cell is going to be very complex. We have a lot of different organelles that we will see on the inside. So just from a visual perspective, you can already see a major difference between prokaryotic cells and also eukaryotic cells. So let's talk about them in a little more depth. Prokaryotes are going to be the simple cell structure. So if you ever seen the word pro, think of like a prototype, right? So like an iPhone 1 is a prototype for something like an iPhone 10, right? It's going to be the early version of it. It's going to be relatively simple. That's what a prokaryotic is. They have a very simple cell structure. Another thing that contributes to their simplistic nature is they do not have a nucleus. So no nucleus will be found in your prokaryotes. Also, they don't have any other type of membrane-bound organelles. So prokaryotes are purely just an open space. Now, they do have some regions in which we will talk about, but they do not have any type of membrane-bound organelles. So once you look inside of a prokaryote, you don't typically have a lot of compartmentalized or sectioned areas. That does not happen in a prokaryote. So let's flip it over and look at the eukaryote. These are going to be very complex cells, right? A lot of our animals and plants are going to be eukaryotic cells. They're complex. How? Because they have their DNA enclosed in this membrane-bound nucleus. So you've heard of a nucleus before. In this image, it's this big kind of central center here, but this is like the brain of the cell essentially, and we're going to have a membrane around it, which will allow it to have its own type of function. And then additionally, we also have internal membranes that can form organelles. So organelles are like a little organ, and we have these internal membranes that are designed to be able to compartmentalize it. So think about your home or wherever you're at right now. Most people's home is going to have separate rooms. So you might have a kitchen, right? In the kitchen, you can do things like cook, you can clean, you can do those aspects. You might also have a living room where you watch Netflix, where you lay out on the couch. You might have a garage. That's where your car is going to go, right? This allows you to do things in the garage that are separately. You have your bedroom. This is where you sleep. This is where you study. All of these things. But because these rooms are all compartmentalized, they all have their separate space. That allows you to do separate tasks and separate functions into each room. That is the beauty of a eukaryotic cell. Because of these compartments, that allows this cell to be able to do different functions, which will make it more complex. So there is a great video about cell biology in these organelles that I'm actually going to show to you in class, so I won't spend too much time here, but it is really important. It kind of gives you an overview of what we're covering. So let's talk about those prokaryotic cells for some time. So prokaryotes are actually going to stem from two domains, okay? The first domain that it stems from is domain bacteria. So these are going to, again, be your small cells, really teeny tiny, 1 micrometer to 10 micrometers in size. Bacteria are super abundant, so we find bacteria everywhere. If you had glasses and could see bacteria through your glasses, you would just be overwhelmed. They're all over the place. And a lot of bacteria truly are not harmful to humans, but of course, some causes diseases that we all know about, bacterial infections, and these kind of get a bad rap. Archaea, like we mentioned before, they're also very small in size, but they're less common. So you do not find archaea everywhere. We only tend to find them in those extreme environments that we talked about. So really, really cold Antarctica, really, really hot places around the equator, those type of concepts. So this is just an image of a bacteria. You see there's a lot of features that you may have seen from images of bacteria, and we're going to kind of cover what those features are. So within a typical bacteria cell, we're actually going to talk about two sides. One is the inside of the plasma membrane, and the second one is the outside. So just to familiarize yourself and remind you a little bit, remember our plasma membrane is made up of those phospholipids. So we talked about phospholipids before, and we discussed the phospholipids have that polar head, that non-polar tail, and we also discussed that they're going to orient themselves in a very particular fashion to form all of our cell membranes. So the membrane that encloses your bacteria, it's going to be made up of that double layer phospholipid membrane. So within that membrane, so in this image here, it is the dark yellow line that you can probably see. It's the closest to the tan interior of this bacteria. Within that plasma membrane, the first thing that you're going to come across is cytoplasm. So cytoplasm is essentially just the entire space that's found within the plasma membrane. So it's that entire region. Please don't confuse this with cytosol, which I will talk about a little bit later. Cytosol is essentially the liquid that you find, while the cytoplasm is essentially the liquid plus all of the solids. That would be your cytoplasm. I have an example a little bit later on in another lecture, but my best example of this is orange juice plus pulp, right? So all of the solid part of the inside of an orange, you have the juice and you have the pulp, versus the cytosol, which is purely just the orange juice, right? It's purely just the liquid. So the cytoplasm includes everything inside the bacteria, while the cytosol is purely just the liquid found within the bacteria. The next portion is called the nucleoid region. So a nucleoid region is where the DNA is at. And remember, these cells do not have a nucleus. So there's nothing surrounding that DNA. It's just kind of a location or a place inside the cell where it tends to be. That is called the nucleoid region. It's a location. And then finally, we have ribosomes. So all those little teeny tiny dots that you see, that is going to be a ribosome. A ribosome is going to synthesize a protein or make a protein. So we talked about this with the term dehydration synthesis. Synthesis means to make. So ribosomes are responsible for making the proteins. So those are the types of components that you see inside of bacteria. You have the cytoplasm, which includes the cytosol. You have a nucleoid region where the DNA is found, and then also ribosomes, which are going to be little teeny tiny areas that we can make our proteins. So we look outside the plasma membrane. So again, our plasma membrane from this image is the dark yellow layer. The first layer outside of the plasma membrane, this goes in order, is the cell wall. So the cell wall is going to be able to provide that support and protection for the bacteria. It's going to aid in the transport of items in and outside of the bacteria. So think of it like being a moat around a castle, right? A circle around a castle. It is going to give it some structure, some protection, also regulate what goes in and outside of the bacteria. And cell walls are found not only in bacteria, but they're also found in plants, and they're made up of that macromolecule that we talked about before, which is cellulose, right? So they're going to be made up of cellulose. The second part in this image is going to be the blue area outside the cell wall is going to be known as the glycocalyx. The glycocalyx. The glycocalyx is going to be used to kind of trap the water around the bacteria so that it always stays moist, and it also stays like in contact with water. And then secondly, it's like a little thick jelly. So this jelly is going to give the bacteria protection. So let's imagine your bacteria bumped up against a cell. This is going to kind of be like a bumper so that it doesn't cause any damage to the bacteria itself. It also helps to evade the immune system. So as certain cells go to attach and bite the bacteria to try to get rid of it, it kind of slips away because this glycocalyx is going to be this slippery external covering that allows a bacteria to be able to kind of slip away from the immune system. So it's a very important component of the bacteria's structure. Thirdly, we have pili. Pili are going to be used for attachment, and there's usually many of them present. So if you see the outside of bacteria, they're going to be little short hair-like projections on the outside that help it to stick. So think of like Velcro, right? If Velcro is made up of little tiny little strings that are going to help it to stick to the other side of the Velcro adhesive. So that pili is going to be useful, right? If you have 1,000 little components and you throw the bacteria against a wall, for lack of better words, all of those components are going to help it to stick. And then finally, flagella. So flagella are found on the outside. They are going to be those long, tail-like projections found on the bacteria, and they are used for movement. So there are very few of them present. You don't have thousands of them. You might have one, you might have maybe 20 or something, but it's not like the pili in which you might have, 3,000 pili on the outside of the bacteria. We do have cells, only one cell in our body or in some people's body that has a flagella, and that will be a sperm cell. So, men have a sperm cell. It has the top part is going to have a lot of the DNA or all the DNA that the male would contribute towards the fetus. And then the tail, that flagella, is going to help that cell travel up the uterus and get to the egg. So we do have one cell with a flagella. So this is a little bit about the structure of the bacteria. This is another image that kind of puts it together. And as you can see here, this is a nice cartoon image with that nucleoid region. You got the ribosomes. Everything inside is a cytoplasm. If we zoom in a little bit, you can see that first blue layer is going to be the plasma membrane, okay? The brown layer is going to be the cell wall, and then the outside yellow layer is the glycocalyx. All of the tiny yellow projections will be your pili, and finally at the very end, those tail-like projections will be your flagella. This is a cartoon image. This is a close-up microscopy image of E. coli. So this is the infamous E. coli. And you can see here some of the components that we talk about from the cartoon. You can also see it real life in this microscopic image. So that gives you a really solid overview about bacteria, about prokaryotes. When we look at eukaryotes, it's going to be a little bit different. So again, remember, eukaryotes, their DNA is found inside of a nucleus. That compartmentalized function is there. So like I said, with a home, you got a kitchen, a bathroom, a garage. All these different areas will allow different things to occur. And then finally, you also have some organelles present. So the organelles are going to be like tiny organs. It literally means little organ. And this is where you have those compartmentalized areas that allow different features to occur. All have different shapes, all have different functions, which we will talk about in depth. And then the final thing here is that variety plays a role. So depending on the cell type, the shape, the size, the different organization will be different because structure leads to function. So as we can change the shape of this cell, this will affect how that cell works. And then also different species will, or different specialization of items in your body will have different types of organelles present, or a difference in the amount of organelles present as well. So this is an image of an animal cell, and as of right now, I know it might look a little overwhelming. There's a lot of components. However, all of these components, or at least a large majority of them, we will cover in the next video. But I do want you to just see the complexity and the beauty of this cell, right? All of the cells in your body are comprised of the same type of component. So while we're able to just walk and talk and move around, all of these little tiny microscopic cells are doing the work, and they're doing the work due to all of these organelles that are present. So this is an animal cell. Of course, this is kind of the main one that we focus on. However, we will talk a lot about plant cells. And I want to show you a picture of a plant cell because besides just the color, there are some different organelles and different components present in a plant cell. I'm going to give you a little hint here and help you to identify some of the organelles that are only found in plant cells as you compare to what is found in an animal cell. And we will do a lot of comparisons in this course between the different types of cells. So a few things that plant cells have that animal cells do not are, I like to think of the three C's. One is a cell wall. Animal cells never have a cell wall. Plants also have chloroplast. And then finally, plants have a structure called a central vacuole. So if you remember those C's, it's going to get you a little bit closer to what those components are. We'll talk about them in depth, but really quickly, the cell wall is going to be this harder exterior layer outside of the cell. The chloroplast is going to be the powerhouse that allows the cell to generate energy from the sun. And then the central vacuole is going to be this big, think of it as like a storage room in which cells can store water so that they can hold the water and use it whenever they need to. So again, just looking between our animal cells and our plant cells, they do have a lot of similar components. Of course, they both have nuclei, they both have endoplasmic reticulum, things like that. However, there are going to be some differences, and I already identified a few of those differences in the plant cells as compared to the animal cells. Just to give you an idea as we close up this video, one thing that we will be doing frequently in this course, especially as it relates to these different cell types, is a number of Venn diagrams. So we will practice them in class. However, this is just an idea. We'll be comparing things. So a Venn diagram shows one side that whatever's on this side is specific only to that term. Whatever's on this side is specific only to that term, and anything in the middle or anything that overlaps, they both have. So this is just, to give you an idea of what to expect. All right, this wraps up this video. Please be on the lookout for the next one.